Hello everyone, welcome to my microtalk. My name is Pan Yufang, and I am from the group of Professor Hu Hanhua in Wuhan, China. I am very happy to be here to share with you our current research. First, let me introduce the TAG biosynthetic pathways. Phaeodactylum tricornutum has the capacity to accumulate TAG which can be enhanced under stress conditions. Two TAG biosynthetic pathways have been clarified: an acyl-CoA dependent pathway and an acyl-CoA independent pathway. The terminal step is catalyzed by DGATs in the former and by PDAT in the latter. Nucleotide variants have been found within alleles of the single PDAT encoding gene in .1 strain, as well as other accessions, by the whole genome resequencing data. The two PDAT alleles are designated as PDAT1 and PDAT2 in this study. PDAT gene is clarified to be allele-specific, expressed in response to environmental stimuli by analysis of previously published RNA-seq data. Besides, the expression of PDAT gene is upregulated during nitrogen and phosphate starvation, which is consistent with the increase in TAG accumulation. Two PDAT alleles were separately overexpressed in P. tricornutum cells. It is indicated that the expression of allele PDAT2 is 3 times higher than that of allele PDAT1 in wild-type cells. Although overexpression of allele PDAT1 resulted in little increase in the TAG accumulation, and overexpression of allele PDAT2 could significantly enhance the TAG contents under nitrogen and phosphate. Limitation and only had minor impact on the growth. 4 reconstructed PDATs, obtained by replacing the fragment in PDAT1 with the corresponding segment from PDAT2, and 2 PDAT alleles were expressed in yeast. The results indicated that PDAT2, but not PDAT1, shows TAG biosynthetic activity in yeast. And the 7 amino acid variants at the N-terminal and the 3 amino acid variants at C-terminal between PDAT1 and PDAT2 had significant effects on PDAT activity. These amino acid variants may have an impact on the folding of PDAT protein. 2 PDAT knockout lines were constructed. Compared with the wild type, they significantly decreased TAG's content and promoted the growth slightly. Comparative analysis of lipid composition was performed in this study, and PDAT knockout strains showed increases in the levels of PC and MGDG, indicating these two lipids were the substrates of PDAT in P. tricornutum. Considering the types of fatty acids that are strongly reduced in TAG and those are correspondingly increased in membrane lipids, a dynamic recycling, mainly of 16:1, is impaired in PDAT knockout strains. Besides, PDAT knockout have contribution to the feeding of membrane lipids with EPA. The maximum likelihood tree of PDAT protein family indicated that P. tricornutum PDAT is homologous to green alga PDATs. The subcellular localization of PDAT is the innermost chloroplast membrane. And this is consistent with the substrate specificity of PDAT in P. tricornutum. Conclusion: 1. The single PDAT encoding gene is allele-specific, expressed in P. tricornutum. 2. PDAT encoded by the more significantly up-regulated allele in response to nitrogen starvation plays an important role on TAG synthesis in P. tricornutum. 3. The innermost chloroplast membrane located PDAT uses membrane lipids as substrates such as MGDG, and shows highest specificity for PC in P. tricornutum. That's all for my microtalk. Thank you very much for your kind attention. If you have any questions, I would be happy to answer them and look forward to your suggestions on my research. Thank you very much.